He kept careful records of all procedures and findings. Controlled pollination between plants with contrasting traits. Generalized are the results of breeding pea plants into what we now know as Mendel's laws of inheritance. Procedure to bring about cross-pollination. White circle the figure. Figure 1 illustrates that the pea plants normally dioecious bisexual, aka having both the androecium and genetium in the same plant, were made by selectively removing androecium to make AFMLE placent. 1. Inheritance biology. Mendelian principles. Introduction. Gregor Johann Mendel, a mathematician and farmer by choice, is known as the father of modern genetics. He studied the inheritance of contrasting characters, traits in pea plants in the Augustinian monastery. Following this, he proposed the laws of inheritance which did not get any recognition until their rediscovery. Many workers verified the laws independently, including Erich von Schermach, Hugo de Vries, and Karl Correns in the 1900s. Mendel implemented scientific ways of breeding pea plants with contrasting characters, which was the key to his success. Some of the key elements of his research are: Figure 1. Illustration of making a monoecious plant refers to the organisms which have both the male and female reproductive organs in the same individual, also termed as hermaphrodite, from a dioecious when the male and female reproductive organs are on different organisms plant. Use of pure breeding line. For elimination of effects of maternal characters, he chose reciprocal crosses. He analyzed a large number of plants quantitative analysis. The environmental conditions were kept the same. He recorded the statistical data very precisely and used the knowledge of mathematics in his experiments. Mendel and his experiments. Mendel was a mathematician with a passion for farming, and it was because of this passion that he began his breeding experiments with pea plants. There were several reasons that led to success in his experiments that others could not achieve till then. Some reasons are outlined below. Using self-fertilized population of pea plant for getting homozygous populations. Choosing the contrasting pair of characters. Figure 2. Figure 2. 7 contrasting pairs of characters studied by Mendel. Some important definitions. Character, an attribute or feature selected for investigation. Figure 2, e.g., skin color, height, etc. Gene. Gene is a segment of DNA molecule that codes for the synthesis of a single polypeptide. It is an inherited factor that determines a characteristic. Figure 3. Allele, an alternative form of a gene that occurs at the same locus on homologous chromosomes, e.g., T and T are alleles for height. Thus, there is a specific place or locus on a chromosome in pea plants which determines the shape of the seed. This locus might be occupied by an allele for tallness T or the one for dwarfness T. Figure 3. Figure 3. The position of allele and gene on homologous chromosomes. Dominant, controlling or being more powerful or important than all others, being reproduced by a form of a gene that prevents or hides the effect of another form. Figure 4. Recessive refers to a trait that is expressed only when the genotype is homozygous. A trait that tends to be masked by other inherited traits yet persists in a population among heterozygous genotypes. Figure 4. Figure 4. Dominant and recessive traits studied by Mendel. Homozygous, when you inherit the same version of a gene from each parent, so you have two matching alleles. Figure 5. Heterozygous, when you inherit a different version of a gene from each parent, they do not match. Figure 5. Figure 5. Homozygous and heterozygous chromosomes. Self-fertilization, fusion of male and female gametes, sex cells produced by the same individual. Self-fertilization occurs in bisexual organisms, including most flowering plants, numerous protozoans, and many invertebrates. Hermaphrodites, organisms in which the same individual produces both sperm and eggs. Many species possess well-developed mechanisms that ensure cross-fertilization. Cross-fertilization, polygamy, fusion of male and female gametes from different individuals of the same species occurs in dioecious plants, male and female organs on separate individuals. Even among figure 6, self-fertilization and cross-fertilization. Reciprocal cross. A cross in which the sex of two parents is reversed is called a reciprocal cross. It is used to test the role of parental sex on a given inheritance pattern. The illustration is shown in figure 7. Figure 7, a reciprocal cross. Test cross. It is an experimental cross of an individual organism of dominant phenotype, genotype unknown, DD or DD, and an organism with a homozygous recessive genotype, DD. Results: white circle if the cross results in all dominant progenies, then the parental genotype must be homozygous. Dominant DD, figure 8. White circle if the cross results in an equal number of dominant and recessive progenies, 1, then the parental genotype must be heterozygous, DD, figure 8. Figure 8, an illustration of test cross. Mendel's success. The main reasons for Mendel's success are outlined below. Method of working. When other scientists were working on an organism as a whole, Mendel decided to choose one character at a time. Mendel carried forward the experiments up to the third generation for a better explanation of inheritance. Being a mathematician, he did the research in a systematic way. Using statistical data was one of his strong points. Homozygous pure lines were developed by self-fertilization for many generations to conduct the cross-breeding experiments. Mendel used a large number of plants for the conclusion of his experiments, which minimized the chances of error. Advantages of selecting pea plant. Pea, being an annual plant, allowed Mendel to study the inheritance of any particular trait for several generations. The short life cycle of pea plants was important in conducting the experiments in a short time and in the same weather conditions, minimizing the environmental effects on the phenotype or genotype. The flowers were complete and could be self-pollinated, hence, it was easy to remove one of the gamete-producing parts to make the pure lines. Other dwarf, for the monohybrid cross. Cross-pollination between tall and dwarf plants produce tall plants, known as F1 progeny. Dominant traits are those that are manifested in the phenotype, whereas recessive traits are those that are not. He then carried on his study of self-pollination in F1 offspring plants. This produced plants that were 3, 1 tall to short, leading to the creation of the law of segregation. Law of segregation. According to Mendel's law of segregation, each gene separates from the others during gamete production, resulting in a gamete with a single allele for each gene. The second inheritance law is the law of segregation. According to this law, a pair of alleles separate from one another during meiosis cell division, gamete creation, resulting in a single allele being present in each gamete. In a monohybrid cross, neither allele is blended, both alleles are expressed in the F2 generation. The fact that each gamete only contains one allele serves as the foundation for the law of segregation. 4 fundamental ideas form the basis of this law. A gene can take on different allele forms. Allelic pairings split apart during meiosis, resulting in each gamete having just one allele. For each trait, an organism inherits 2 alleles. A pair of alleles has 2 distinct alleles, one of which is dominant and the other recessive. Law of independent assortment. According to the rule of independent assortment, sexually reproducing organisms inherit distinct alleles of genes independently. The law of independent assortment. The alleles of 2 additional genes are sorted into gametes independently of one another in accordance with the law of independent assortment. Receiving one allele for one gene does not affect receiving another allele for a different gene. There were a lot of evident contrasting pairs of characters which could be explained as dominant and recessive. A lot of progeny seeds were produced per generation in pea plants. Gregor Mendel established the fundamental laws of inheritance while working on pea plants. He deduced that genes come in pairs and are passed down from one parent to the other as separate units. Mendel studied the segregation of parental genes and how they manifested as dominant or recessive traits in their offspring. He was familiar with the mathematical patterns of inheritance passed down from generation to generation. Mendel's law. Law of dominance. According to Mendel's law of dominance, only one type of trait manifests in the offspring of parents who possess distinct, opposing traits. The sole phenotypic trait that the hybrid offspring will display is the dominant trait. The first law of inheritance is known as the law of dominance. Each character in this law is governed by individual, paired units known as factors. In heterozygous couples, one will always prevail over the other. According to the law of dominance, only one of the contrasting features from the parents will be displayed in the F1 generation, and both parents' traits will be expressed in the F2 generation in a 3:1 ratio. A recessive trait is one that is suppressed, while a dominant trait is one that is displayed in the F1 generation. The law of dominance essentially argues that the dominant trait always outweighs or obscures the recessive traits. The Mendel experiment can be used to explain this law. A monohybrid cross is created when 2 monohybrid traits are combined TT and TT. Plants that were crossed in this instance share the same characteristics but varied by just one character. Mendel started with a pair of pea plants that had 2 distinct features. One tall and the parent passes an allele at random to the offspring resulting in a diploid organism. The allele that contains the dominant trait determines the phenotype of the offspring. Figure 9. In essence, the law states that copies of genes separate or segregate so that each gamete receives only one allele. Possible outcomes. 3 types of genotypes were observed by Mendel in the F1 generation. Homozygous dominant. Heterozygous homozygous recessive. Mendel's experiment consistently shown that the progeny's qualities are always distinct from those of their parents. He developed the law of independent assortment on the basis of this. Mendel's law of dominance. A diploid individual possesses a pair of alleles for any particular trait, and each parent passes one of these randomly to its offspring. Observation. Crossing true breeding pea plants with contrasting traits F1 generation all dominant. Selfing F1 dominant and recessive 3. 1 generalization. Mendel proposed the law of segregation. Each individual that is a diploid has a pair of alleles copied for a particular trait. Each figure 9. Law of segregation. Physical basis of segregation. Mitosis and meiosis were first described in the late 1800s. The chromosome theory of inheritance states. White circle Mendelian genes have specific loci positions on chromosomes. White circle chromosomes undergo segregation and independent assortment. The behavior of chromosomes during meiosis can account for Mendel's laws of segregation and independent assortment. Figure 10. Figure 10. Physical separation segregation of chromosomes during the anaphase stage of meiosis suggests the independent assortment of chromosomes. Law of independent assortment. The law of independent assortment suggests that the unlinked separate genes for separate traits inherit individually or independently from one generation to the next. There is no influence of genes of one trait in assorting alleles into gametes of the other traits taken into consideration, and there may be a probability of any combination of the alleles for every gene. To calculate the possibility of certain genotypic combinations in two or more genes, white circle multiply the probabilities of a certain genotype at first and the other loci. White circle add the probabilities of a certain genotype one or more type states of either this or that at first and the other loci. Dihybrid cross is a classic example of the law of independent assortment. New combinations of traits appeared in the F2 generation when the selfing of heterozygous brown and yellow F1 progeny was done. New traits were round green and wrinkled yellow, which suggested the independent assorting of the two different traits, as depicted in Figure 11. Figure 11. Dihybrid cross law of independent assortment. The expected distribution of phenotypes and or genotypes is represented by the branch point for a particular trait. The expected fractions can be multiplied for calculation of the expected frequency. Figure 12. Fourth line method. Dihybrid cross. The fourth line method is used to figure out the occurrence of possible phenotypes and genotypes in 3 or more unlinked genes. Figure 12. Fourth line method in a dihybrid cross. Fourth line method. Trihybrid cross. Illustrated in Figure 13. Figure 13. Fourth line method in a trihybrid cross. Exclusive events is the sum of their individual probabilities. Product rule. The probability of two independent events occurring together can be calculated by multiplying the individual probabilities of each event occurring alone. Binomial theorem. xn p x p q n x x minus equals minus where n equals the number of trials or the number being sampled. X equals the number of successes desired. P equals probability of getting success in one trial. Q equals 1. P equals the probability of getting a failure in one trial. Probability. The rules of probability can be applied to Mendelian process to determine the expected phenotypes and genotypes of the offspring. Key points. White circle genetics depends on chance factors. White circle, we have an entire genome in which gene is going to get expressed as completely a chance factor. So, we use a mathematical technique called rule of prediction or probability. Probability of an event which is 100% possible, right pointing arrow 1. Probability of an impossible event, right pointing arrow 0. Rules of probability. Sum rule. Product rule. Binomial theorem. Sum rule. Additive rule. The probability of the occurrence of one event or the other event of two mutually. Mendel was a mathematician, but he worked on pea plants and gave the laws of inheritance. There were so many things he took into consideration while studying the inheritance pattern of the pea plant, and many reasons are discussed in the chapter. He postulated 2 laws: law of segregation and law of independent assortment. Chapter summary. 2 concept of gene. Definition. Each gene, according to Gregor Mendel, should contain just 2 alleles. Alleles are referred to as gene variants that come in 2 or more forms. Each gene is inherited from each parent in 2 alleles. This implies that a trait would also have 2 distinct alleles. Children with genotypes made up of the same alleles, i.e., of a gene for a specific trait, are said to be homozygous, whereas those with different alleles of a gene are said to be heterozygous. Genetically, humans and all other diploid species can only have 2 alleles for any particular gene. However, at the population level, numerous allele traits may occur. As a result, numerous alleles play a crucial role in encouraging variety within a single species. A specific chromosomal location in Mendelian inheritance was home to 2 unique sorts of gene alternatives, one dominant and one recessive. 2 alleles of the same gene make up these alternatives. There are, however, some situations in the population where there are more than 2 choices alleles. These situations, where several variants of the same gene occur in the population, are known as multiple allelism. Multiple alleles are these 3 or more variations of the same gene. They are always at the same locus of the chromosome. Figure 1. Introduction. The definition and concept of a gene is not limited but continuously evolving. From classical times, one can comprehend the concept in the 1930s, where a gene was defined as the inseparable unit of mutation, genetic transmission, function, and recombination. In neoclassical periods, 1940s to 1970s, DNA was established as the physical basis of inheritance after the discovery of intergenic recombination. The division of the gene into the functional units of cistrons, 1 gene and 1 polypeptide, mutons, 1 mutation, and one recombination, and identification of the nucleotides that make up each of these units followed. The collinearity theory was created because 1 cistron in a gene can produce 1 mRNA and ultimately 1 protein. Neoclassical period, 1955 to 1970s. In modern views, from the early 1970s, DNA technologies were used tremendously, which defined the gene in new perspectives: repeated genes, interrupted genes and alternative splicing, overlapping genes, transposons, jumping genes, assembled genes, promoters, untranslated regions, UTRs, polyannulation sites, post-transcriptional modifications, nested genes, and so on. Therefore, a gene is an evolving concept, and we do not know what future technologies can reveal about it. Figure 1. Gene loci for different traits are shown in the figure. There is no crossing over within the alleles of the same multiple allelic series. Figure 2. Figure 2. Recombination in homologous chromosomes. Crossing over can only result in intergenic recombination between 2 different genes, and not intergenic recombination within a gene. Multiple alleles. Sometimes, more than 2 alleles may be responsible for different phenotypes in a species. This is termed as multiple allelism. It is a non-Mendelian form of inheritance. The wild type allele is usually dominant over all others at the same locus. When 2 mutant multiple alleles are crossed, the phenotype is mutant type and not wild type. Multiple alleles always influence the same character. They control the same character, but each of them is characterized by a different manifestation. Examples of multiple alleles: Drosophila eye color. Figure 3. Figure 3. Drosophila eye color. Rabbit coat color. Figure 4. Figure 4. Rabbit coat color. ABO blood group system in humans. Figure 5. Figure 5. ABO blood group system in humans. Identifying the genotype number kinds when the number of alleles is given. The number of genotypes, homozygotes and heterozygotes, can be calculated as per the following formulae. Table 1. White circle n1, number of kinds of heterozygotes if in alleles are present. 2. Plus equals. White circle, number of kinds of homozygotes if in alleles are present equals n. White circle n1, number of kinds of heterozygotes if in alleles are present. 2. Minus equals. Number of alleles, genotypes, homozygotes, heterozygotes 1 1 1 0 2 3 2 1 3 6 3 3 4 1 0 4 6 5 1 5 5 1 0 n. Table 1 kinds and numbers of the genotypes. Incomplete dominance. The situation in which the phenotype of a heterozygote is immediate between the 2 homozygotes on some quantitative scale of measurement is called incomplete dominance. Figure 6. In the absence of complete dominance, every genotype has a distinguishable phenotype. Figure 6. Incomplete dominance and its inheritance. It is also termed semi-dominance because alleles may produce the same product but in lesser quantities as compared to the dominant allele. In the heterozygous condition, the total product is intermediate between that of the dominant and recessive allele. Figure 7. Figure 7. Incomplete dominance, a case of haploid insufficient allele. Phenotypic ratio for monohybrid crosses between 2 heterozygous results in a 1 2 1 ratio instead of 3 1. Figure 6. Molecular explanation. In incomplete dominance, only one allele heterozygous is expressed to produce a product. When a single copy of the wild-type allele is present at a locus in heterozygous combination with a variant allele, the wild-type phenotype cannot be produced, according to the half-point sufficiency model of dominant gene action in genetics. A loss-of-function mutation in the variant allele, which results in it producing little to no gene product, can cause half-point sufficiency, often of protein. The total amount of product is not enough to result in the expected phenotype, even while the other, conventional allele still generates the expected amount. This heterozygous genotype may lead to a disease phenomenology that is subpar, non-standard, or harmful. Most commonly, dominant harmful alleles are explained by half-point sufficiency. Genetic basis. It may be due to the differences in the amount of product specified by the dominant allele. Figure 8. Figure 8. The amount of gene product in incomplete dominance may be the reason for intermediate phenotype. Gene is half-point sufficient. Lethal genes. The phenotypic manifestation of some mutant genes results in the death of the individual, either in the prenatal or postnatal period, prior to sexual maturity. Such genes are called lethal genes. Figure 9. Figure 9. Lethal genes. Here, genetic defects cause 100% mortality. They are not limited to X chromosomes. They frequently occur in the autosomes. It involves the interaction of many genes. It might interfere with the development of some vital organs, e.g., heart, or it might upset some vital processes, chemical or physiological, early in development. Types of lethal alleles: white circle dominant lethal, white circle recessive lethal, white circle completely lethal, white circle semi-lethal, white circle conditional lethal, white circle synthetic lethal, dominant lethal. Individuals with dominant lethal allele die before they can leave behind any progeny. Therefore, the mutant dominant lethal allele is removed from the population in the same generation in which it arose. More lethal effects occur in heterozygous condition. Example: Huntington's disease in humans. Dominant traits can be maintained in the population through recurrent mutations or if the penetrance of the genes is less than 100%. The slow onset of Huntington's disease causes it to pass the gene through a population. Recessive lethal. With the snapdragon plant Andreinum, Erwin Bauer started his research in 1907 and described the condition Rhea, which causes plants to have golden leaves rather than green flower. 1907, Castle and Little, 1910. Bauer saw in achondroplasia are few conditions that are brought on by recessive lethal alleles in humans. Dwarfism is a bone condition caused by achondroplasia, an autosomal dominant gene. Two recessive lethal alleles are required for death, whereas one achondroplasia allele can result in the disease. It kills only the homozygotes. Genotype phenotype LL, LI normal viability. LL lethal. Table 2 recessive lethal kills only in homozygous form. Figure 9. A 2:1 ratio of green to yellow seedlings when two Rhea snapdragon plants were crossed. Homozygous Rhea plants lacked normal chlorophyll formation and perished either in the embryonic stage or 2 to 3 days after plant seedling emergence. In other words, like Quinott's homozygous mice, the homozygous Rhea plants were unable to fully grow, which resulted in the death of an entire class of offspring. Castle and Little, 1910. Because they changed Mendelian inheritance ratios, Quinott and Bauer found these first recessive lethal genes. It is possible for recessive lethal genes to code for dominant or recessive features, but only if an organism possesses 2 copies of the lethal allele they really result in death. Cystic fibrosis, sickle cell anemia. Figure 9. Lethal gene inheritance exhibiting a distinctive phenotype in heterozygotes. Examples. In snapdragon Andreinum, the amount of chlorophyll is controlled by an incompletely recessive gene which exhibits a lethal effect in homozygous state and a distinctive phenotypic effect in the heterozygous state. Table 3 and Figure 10. Genotype phenotype. CC green normal with chlorophyll. CC golden for Rhea, yellowish green with carotenoids. CC white lethal and without chlorophyll. Table 3 recessive lethal gene in snapdragon. Figure 10. Recessive lethal gene in snapdragon. Completely lethal. It usually causes death of the zygote, later in embryonic development or after birth. Figure 11. Here no individual of a certain genotype attains sexual maturity. The lethal genes have a high degree of penetrance and expression. However, in many cases, lethal genes permit larger proportions of affected genotypes to survive. White circle such genes are called subvital, sublethal or semi-lethal. Semi-lethal. Genes that kill fewer than 100%, but more than 50% of the victims are called semilethal. White circle subvital. Genes that cause less than 50% mortality are known as subvital. White circle supervital. Some mutant alleles enhance the survival of those individuals that carry them in the appropriate genotype as compared to that of wild-type allele. Such genes are known as supervital. Examples of lethal genes. Yellow, lethal in mice. Figure 11. Figure 11. Yellow lethal in mice. In Drosophila. Examples. Curly wings, upward bending of wings. Figure 12. Plum eyes, eye color. Dickey absence of certain bristles and spread apart wings. Figure 12. Curly wings in Drosophila is an example of recessive lethal genes. Creeper fowls. Figure 13. Figure 13. Inheritance of creeper fowl lethal gene. In humans. Congenital ichthyosis, leathery skin with deep fissures. Thalassemia, Cooley's anemia. Figure 14. Amauroid idiocy. Figure 14. Lethal gene inheritance of thalassemia. Figure 15. Himalayan rabbit, black color and extremities otherwise white. The typical phenotype is a result of an allele that produces a temperature-sensitive gene. Mutation in this gene has caused a change in the structure of tyrosinase, so it works only at low temperatures, hence it works only in cooler regions of the body. Rabbit is endothermic and maintains its body temperature 34 OC in core areas of the body. Conditional lethal. Definition. Conditional mutation is a mutation that has a wild type or less severe phenotype under certain permissive environmental conditions and a mutant phenotype under certain restrictive conditions. Characteristics. Conditional mutations are also termed non-autonomous because their expression depends upon certain conditions. The permissive conditions may be temperature, certain chemicals, lighter mutations in other parts of the genome. Temperature-sensitive mutants are best suited to analyze the function of such genes, where mutation in gene leads to lethality in a haploid organism. Examples: white circle Himalayan rabbit is depicted in figure 15. Figure 16. An experiment was conducted for the testing of temperature's effects on a rabbit as shown in the picture. An ice pack was kept on the back of the rabbit which resulted in color expression on the back of the Himalayan rabbit. The concept of gene can be defined in different ways and has been evolving from the classical to the modern period of development of genetics. Traits can be characterized by multiple alleles or lethal genes. A lot of different traits are inherited in humans and other organisms because of these different types of interactions as discussed in the chapter. Chapter summary. Extensions of Mendelian. Principles 3. Figure 1. Codominance is alleles in heterozygote expression inheritance. Pseudoallele. Definition. These are closely linked alleles, arranged linearly in a complex locus consisting of different subloci within which crossing over can occur. They are structurally non-allelic and functionally allelic and they show cis transposition effect. Introduction. Mendel was fortunate in choosing the traits in pea plants which exhibited a rather simple inheritance pattern, as dominant and recessive, for a single trait. However, numerous inheritance patterns, most do not follow the single gene concept as Mendel's work depicts. There are some alternatives to the single gene concept, dominant and recessive allele, which are discussed later in the chapter. Codominance. Alleles that lack dominant and recessive relationship and both have the capability to express themselves phenotypically in heterozygous conditions are termed as codominant alleles. It exhibits a unique pattern of expression with heterozygotes being phenotypically distinguishable from both of the homozygotes. Here, two alleles appear to contribute independently to the phenotype of the heterozygote. Here, both alleles are expressed equally and simultaneously. In general, two products for the same WRT function but are different in amino acid sequence. Codominant alleles are represented by superscripts on the symbol for the gene. Examples: coat color of short-horn breed of cattle. Figure 1. Characteristics. These are closely linked alleles within which crossing over can occur. Hence, it is structurally non-allelic in nature. They affect the same character, so they are allelic in function. These alleles are arranged in a linear order within complex loci. This complex locus is divisible into several sublocus. Pseudololes of the same sublocus do not undergo recombination. Those alleles occupying different sublocus undergo recombination. Pseudololes have cis transposition effect. When they are in the cis configuration, they show wild type phenotype, and when they are in trans configuration, mutant phenotype appears. Figure 2. Figure 2. Cis trans effect in pseudololes. Example: garnet locus in Drosophila. New wild type may rise through recombination among garnet alleles G1, G2. Recombination is 0.003%. 1. Female heterozygote of genotype Sable S and Garnet G1 on the X chromosome and Garnet G2 and pleated place on the other X chromosome. 2. Sable is at locus 43.0 and lies to the left of Garnet locus 44.4, while pleated lies to the right of the Garnet at locus 47.9. 3. Since heterozygous females also carry wild type allele for Sable and pleated, crossing over results in female wild type, i.e. S++ place figure 2. Example, lozenges in Drosophila, figure 3. Figure 3, lozenges in Drosophila. Gene interaction definition. When two different loci affect the same characteristics, the outcome of dihybrid cross involving these genes can produce a phenotypic ratio very different from 9, 3, 3, 1. Collaborator genes. When two genes residing at two loci interact and produce a different phenotype for a character, as compared to when a single gene is yielding a phenotype, it is termed a collaborator gene. Examples, white circle fruit color in pepper red, brown, yellow, or green figure 4. Figure 4, collaborator genes pepper fruit color inheritance white circle cone shape in chickens figure 5. Figure 5, cone shape in chickens. Types. Recessive dominant. Recessive epistasis. In recessive epistasis, epistatic gene influences the phenotype of hypostatic gene only when it is recessive. For e.g., coat color in Labrador dog, table 1, coat color in mice figure 6. Epistasis. When one gene masks hides the effect of another gene at a different locus, it is known as epistasis. The gene that is the masking epistatic gene. The gene whose effect is masked hypostatic gene. Figure 6, coat color in mice. Genotypes ratio phenotypes. Observed. Phenotypic ratio. F2A_B_916 black 9AB_316 brown 3A_BB316 yellow 4ABB116 yellow. Table 1 recessive epistasis. Dominant epistasis inhibitory genes. In dominant epistasis, epistatic gene influences the phenotype of hypostatic gene only when it is dominant. For e.g., fruit color in summer squash, Cucurbita pico, Fig. 7 and Table 2. Figure 7, dominant epistasis. Genotypes ratio phenotypes observed phenotypic. Ratio F2A_B_916 white 12AAB_316 white. _BB316 yellow 3AABB116 green 1 Table 2 dominant epistasis. Complementary genes duplicate recessive epistasis. In complementary gene interaction or duplicate recessive epistasis, measurable phenotype is observed only when at least one dominant allele is present at both loci. Phenotype is affected if either the dominant gene or the hypostatic gene is recessive. For e.g., albinism in snails figure 8. Figure 8, albinism in snail. Dominant suppression epistasis. Example, Malvidin production in Prenula anthocyanin giving blue flower figure 9 figure 9. Malvidin production in Prenula anthocyanin giving blue flower. Malvidin production is controlled by a dominant allele at K locus, but the pathway is blocked by a dominant allele at the suppressor locus. C. This produces 13, 3 ratio, like feather color in chickens. Duplicate dominant epistasis. Example, seed capsule shape in shepherd's purse, Capsella bursa pastoris figure 10, duplicate dominant epistasis. A dominant allele at either of the two loci produces a heart-shaped fruit. Total dominance of both gene pairs, but when one gene is dominant, it masks the effects of the other gene. Duplicate interaction. Example, fruit shape in summer squash figure 11. Figure 11, fruit shape in summer squash duplicate interaction. Complete dominance of both loci, but ABGI is a new phenotype as does AABB. This is also called a cumulative duplicate effect and gives a 9, 6, 1 ratio. Summary of gene interactions is depicted in the figure 12. Figure 12, summary of gene interactions. Pleiotropy. Definition and examples. It is a phenomenon in which a single gene contributes to multiple phenotypic ratios. Figure 13. Examples. Fruit flies vestigial gene. Chicken frizzle trait pigmentation and deafness in cats. Figure 13, pleiotropy. Inheritance, which identify chromosomes as the genetic material responsible for Mendelian inheritance. Observations consistent with Mendel's law. During meiosis, homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. The sorting of chromosomes from each homologous pair into 3 gametes appears to be random. Each parent synthesizes gametes that contain only half of a chromosomal complement. Even though male and female gametes, sperm and egg, differ in size and morphology, they have the same number of chromosomes and suggesting equal genetic contributions from each parent. The gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. Eye color in fruit flies was the first X-linked trait to be discovered. Thus, Morgan's experiments with fruit flies solidified the chromosomal theory of inheritance. Fruit fly in vestigial wing. The vestigial gene plays a critical role in wing development. BB will develop short wings. The effects are: white circle, the individual cannot fly. White circle, the number of egg strings in a fly's. Ovaries changes white circle, the position of bristles on a fly's. Scutellum alters white circle, the length of a fly's life decreases. Linkage and crossing over. Chromosomal theory of inheritance. Principle. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publications and reevaluate his model in terms of the behavior of chromosomes during mitosis and meiosis. In 1902, Theodore Boveri observed that proper embryonic development of sea urchins does not occur unless chromosomes are present. That same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. Together, these observations led to the development of the chromosomal theory of Figure 14, recombination of chromosomes. Linkage. When two or more genes are located in the same chromosome, they are said to be linked as they are physically linked or joined together by the segment of chromosome between them. Figure 15. It indicates white circle genes are linked. White circle alleles of the genes are linked. White circle gene loci are linked. Figure 15, concept of linked and unlinked genes. Linkage phase. The arrangement of genes in a particular genotype is called linkage phase. The genes on the same chromosome are said to be in coupling phase, while those on the opposite chromosome of the pair are in repulsion phase. Figure 16. White circle cis heterozygotes, A and B, in one chromosome and AB in another chromosome of the same homologous pair. Figure 16. White circle trans heterozygotes, A and B, in one chromosome and A flat in the other chromosome of the same homologous pair. Figure 16. Figure 16, coupling and repulsion phase of linkage. It is also known as a crossover map because this map is generated by estimating the number of times a crossover event occurred in a particular segment of a chromosome out of all meiosis examined. Linkage map. The diagrammatic graphical representation of relative distances between linked genes of a chromosome is called a linkage map or genetic map. Figure 17. Figure 17, linkage map, genetic map versus cytogenetic and physical map of genes. Significances. This map displays exact locations, arrangements, and combinations of genes in a linkage group or chromosome. It shows the number of linkage groups of an organism. It shows the relative distance and chances of crossing over between the genes. They are useful in predicting the results of dihybrid and trihybrid crosses. Determination of map distance. The distance between two given genes is calculated according to the percentage of crossing over because crossover frequency is directly proportional to the distance between genes. Figure 18. This distance is measured in map units mu. A crossover frequency of 1% between two genes is defined as a map unit. It is also expressed as Morgan unit, i.e., 100% crossing over. The map unit is sometimes referred to as centimorgan cm, i.e., 1% crossing over, in honor of geneticist T.H. Morgan, Nobel laureate. 10% crossing over is referred to as 1 decimorgan. Although the mu is not a physical distance, it is a relative distance that makes it possible to know the order of and relative separation between loci on a chromosome. Figure 18, types of crossing over and its recombination frequency, RF. Cytological or physical map. The physical location of a gene in a chromosome which is accomplished by identifying a gene locus in relation to some visible landmark, viz., chromomer, or crossband, is termed as a physical or cytological map. Linkage map versus physical map. The frequency of crossing crossover usually varies in different segments of the chromosome, but it is a highly predictable event between the two gene loci. The actual physical distance between linked genes bears no direct relationship to the linkage map distances. The linear order of genes is identical in both cases. Figure 19. Figure 19, physical map versus linkage map. Linkage group. All the genes which are linked with one another constitute a linkage group and tend to be inherited together. The number of linkage groups in a species is equal to the number of homologous pairs of chromosomes or the haploid number of chromosomes of that species. Organism number of pairs of homologous. Chromosome number of linkage group. Drosophila 4 pairs 4. Human 23 pairs 24 in males X and Y chromosomes. 23 in females X and X chromosome, maize 10 pairs 10. Table 3 number of linkage groups in some organisms. Types of linkages. Linkage may be of two types. Sex linked. Autosomal. Complete incomplete. Complete linkage. In this type of linkage, genes are closely associated and tend to transmit together upon coming from the same parent for two or more generations in a continuous and regular fashion. Examples: white circle, the Drosophila 4 chromosomes genes for bedwings and shaved bristles show full linkage. White circle, these two gene pairs have an extremely strong linkage since there is essentially no independent assortment between them. Figure 20, complete linkage. Incomplete linkage. The linked genes which are widely located in chromosomes and have chances of separation by crossing over are called incompletely linked genes, and the phenomenon of their inheritance is called incomplete linkage. The linked genes do not always stay together because homologous non-sister chromatids may exchange segments of varying lengths with one another during the meiotic prophase. This sort of exchange of chromosomal segments in between homologous chromosomes is known as crossing over. Example: incomplete linkage has been reported in female Drosophila, figure 21, and various other organisms such as tomato, maize, pea, mice, poultry, and man. Figure 21, incomplete linkage. Significance of linkage. The phenomenon of linkage has one of the greatest significance for living organisms in that it reduces the possibility of variability in gametes unless crossing over occurs. Crossing over. Crossing over meiotic recombination occurs during prophase figure 23, of meiosis at the bivalent stage, zygoting, and completes in three stages: zygoting, packaging, diploiding. Non-sister chromatids of homologous chromosomes exchange DNA segments during this process. Meiosis is shown in the figure 22. Figure 22, stages of meiosis I and II. Figure 23, recombination occurs in prophase I. It can be used as a basic genetic map. White circle, average number of chiasmida per cell equals 210 100 equals 2.1. White circle, average number of chiasmida per chromatid equals 2.1. 2 equals 1.05. White circle, genetic distance of chromosome equals 1.05. Morgan's equals 105 cm. White circle, relationship between genetic length and average number of chasma equals 105 halves. 1 equals 50 cm chasma. White circle, we can conclude that on average each chasma corresponds to 50 cm on the genetic map. Significance of crossing over. Crossing over is universal in occurrence and occurs in plants, animals, bacteria, viruses, and molds. Meiotic crossing over allows a more independent selection between the two alleles that occupy the positions of single genes, as recombination to FFLES, the allele content between sister chromatids. Helps improving linear arrangement of genes. Recombination does not have any influence on the statistical probability that another offspring will have the same combination. This theory of independent assortment of alleles is fundamental to genetic inheritance. Origin of new character. Necessary for natural selection, as it increases the chances of variation. Genetic maps with two-point test cross. Genetic maps can be constructed by conducting a series of test crosses between pairs of genes and examining the recombination between them. A test cross between two genes is called a two-point cross or a two-point test cross. Suppose that we carried out a series of two-point crosses for four genes, A, B, C, and D. The resulting recombination frequencies. Features of crossing over. Occurs at two levels: the gross chromosomal level, chromosomal recombination, and the DNA level, genetic recombination. Occurs between non-sister chromatids of homologous chromosomes. Exchange is normally reciprocal but sometimes unequal. Frequency of crossing over is closely related to the physical distance between genes located on chromosomes. Factors controlling crossing over. Age. Sex. Temperature. Radiations. Chemicals. Physical distance between genes. No. Of. Chiasmida. A number. Of cells observed. B. Product. A times B. 0100125235703103041030415605525. 100,210 table 4 chasma frequency and genetic map distance. Inference. Recombination between the genetic marker is an outcome of crossing over. Each chasma is thought to represent the resolution of a crossing over. The average number of crossovers occurring on a chromosome can be estimated by counting the chiasmida. Gene pairs recombination. Frequencies in percent. A and B50. A and C50. A and D50. B and C18. B and D15. C and D26. Table 5 experimental data is given for preparing two-point test cross. Figure 24. The three steps of determining a genetic map. The RF between A and B is 50%, expected to be on a different chromosome or very far, shows independent assortment. Figure 24a. The RF between A and C is 50%. These two, present on different linkage groups. B and C equals 18%. Hence these are linked and separated by 18U. Figure 24b. The RF between A and D is 50%. These two, present on different linkage groups. B and D equals 15%. Hence these are linked and separated by 15U, but it could be present to the left or right to the gene B. For that RF of C to D is also needed. Figure 24c. The RF of C and D is 26. Hence these are far. Therefore, B should be in the middle, and D should be to the left of B. 3-point cross and linkage. Figure 25. Analyzing three genes allows us to determine gene order as well as linkage distance. One needs to create an F1 and follow deviation from a 1-1-1-1-1-1-1 ratio. What are the expected gametes when three linked genes are considered? Figure 25. Parental and recombinant gametes derived from 3-point cross. Genotypes observed. Types of gamete: ABC 390 parental, ABC 374 parental. ABC 27 single crossover between gene C and B. ABC 30 single crossover between gene C and B. ABC 5 double crossover. ABC 8 double crossover. ABC 81 single crossover between genes A and C. ABC 85 single crossover between genes A and C. Total 1000. Table 6 analyzing a 3-point test cross data. What are the parental genotypes? The genotypes most frequently found are the parental genotypes. ABC and ABC are the parental genotypes. What is the gene order? The double crossover moves a non-parental allele of the central gene between two parental alleles. Gene C is between genes A and B. Gene order equals ACB. What are the linkage distances? Linkage distance equals the sum of the appropriate single cross plus all double crosses divided by the total number of gametes. AC equals 100 * 81 plus 85 plus 5 plus 8. 1000. Linkage distance equals the sum of the appropriate single cross plus all double crosses divided by the total number of gametes. AC equals 100 * 81 plus 85 plus 5 plus 8. 1000 equals 17.9 cm. equals 100 * 27 plus 30 plus 5 plus 8. 1000 equals 7.0 cm. Example. Determine the parental genotypes. Genotype observed. Type of gamete: BCB plus court plus 580 parental. B plus CB court 592 parental. BCB court plus 45 single crossover between genes court and CB. B plus CB plus court 40 single crossover between genes court and CB versus CB court 89 single crossover between genes C and court. B plus CB plus court plus 94 single crossover between genes C and court. BCB plus court 3 double crossover. B plus CB court plus 5 double crossover. Total 1448. Table 7 3-point cross data. Parental genotypes. The most abundant genotypes are BCB plus court plus and B plus CB court. Determine the gene order. Allele court is paired with B and CB plus. This is a different pairing than the parental genotype. Therefore, gene court is in the middle. Determine the linkage distance. B chord equals 100 asterisk 89 plus 94 plus 3 plus 5 1448 equals 13.2 cm chord CV equals 100 asterisk 45 plus 40 plus 3 plus 5 1448 equals 6.4 cm interference. Interference means the tendency of one crossing over to interfere with the occurrence of another crossing over in its nearest vicinity or adjacent region. The phenomenon of Chasm interference or chromosome interference was first observed by H. J. Muller, 1916. The degree of interference may vary in different regions of the chromosome. It is found to be inversely proportional to the distance between the points of crossing over. It means interference is maximum over a short distance and decreases as the distance between the two crossing over points increases. It is maximum near centromere and telomere. In Drosophila, interference prevents DCO between the genes that are 10 to 15 U apart. The degree of interference is measured by the coefficient of coincidence COC. Observed DCO coc. Expected DCO equals as the ratio between the observed actual number frequency of DCO and the expected number of DCO. Observed DCO coefficient of coincidence. Expected DCO equals. It is the complement of interference, i.e., coincidence plus interference equals 1.0. Coincidence becomes 0 when interference is complete, 1.0, and therefore no DCO. Coincidence values ordinarily vary between 0 and 1. If the coefficient of coincidence is larger than 1, it indicates that the occurrence of DCO is more frequent than expected. Coc frequency of DCO. Types of interference. 0 if DCO equals 0% absolute. And LT, 1 DCO less frequent than expected. Partial. 1 observed DCO equals expected DCO. None. And GT, 1 DCO more frequent than expected. Negative. Table 8. Sometimes the level of interference is given as the coefficient of interference. Coefficient of interference equals 1 COC interference types. Positive interference. Positive interference indicates that one crossing over interferes with the occurrence of a second crossing over nearby. Interference value between 0 and 1 indicates that positive interference partial has occurred. Interference is 100% when DCO is altogether absent. If the frequency of DCO is equal to the expected value, it means that there is no interference, none. It is generally observed in eukaryotes. Negative interference. Negative interference indicates that the occurrence of one crossover increases the likelihood of additional crossovers occurring in the nearest vicinity clustering of crossing over. It occurs in certain microorganisms, particularly in bacteriophages, fungus, aspergillus, etc. Coincidence. The coefficient of coincidence is the inverse measure of interference and is expressed. In this chapter, numerous types of gene interactions and concepts were discussed which do not follow Mendel's law of inheritance. There were different ratios of phenotype or different types of phenotypes were observed during the course of breeding different organisms with different characters. The interactions discussed were collaborator genes, complementary genes, epistatic genes, supplementary genes, pseudogenes, linkage, and many more. The chapter contains a brief description of the inheritance pattern in each case with all the essential figures and tables. Chapter summary. 4-gene mapping method. However, when the meiotic products are not arranged in linear order, they are termed unordered tetrad, yeast, or plummononas. Meiosis occurs in all sexually reproducing organisms, but in most of them, the products of meiosis are mixed, such as in the case of pollen grains. However, in a few cases, the products of a single meiosis are held in a separate body. It is possible to isolate each such spore-containing body, which can be further dissected to isolate and culture each spore separately. Because one can isolate the meiotic products and grow them too, the study of their phenotype and genotype is also easy. Y haploid system is suitable for genetic mapping. Higher organisms that undergo meiosis are diploids or polyploids. Some lower organisms which undergo meiosis spend a considerable part of their life cycle in a haploid state. The haploid organisms provide a suitable system since there is only one allele in the haploid state and there is no dominance phenomenon. Unordered spores in yeast. Saccharomyces cerevisiae can exist in two forms, haploid and diploid. The haploid form usually forms under nutritional stress. When better nutritional conditions come, the haploid cells of the two sexes and alpha-mating types fuse to form the diploid. Figure 1. Introduction. Gene mapping refers to the technique of locating genes on a chromosome. Gene mapping can be of two types, physical mapping and genetic or linkage mapping. The concept of linkage is required for the determination of the location of two linked genes present on a single chromosome. The closer the two genes are located on a chromosome, the greater will be the probability of them being inherited together. Using this knowledge, we can establish a relative distance between them. The genetic linkage mapping can also provide us with strong evidence about the inheritance of any disease from one generation to another. Haploid mapping tetrad analysis. A paired homologous chromosome is called a tetrad as 4 chromatids can be observed. In tetrad analysis or haploid mapping, one can use the product of a single meiosis event to locate the genes of haploid eukaryotic organisms. Tetrad analysis involves analyzing the 4 meiotic products and is carried out in some haploid organisms such as yeast, Chlamydomonas, and Neurospora. There are some fungi, e.g., yeast and Chlamydomonas, in which tetrad contains 4 ascospores, whereas some others, e.g., Neurospora spp, have 8 ascospores. Ordered and unordered tetrad. When the spores are arranged in linear orders for obtained meiotic products, they are termed ordered tetrad Neurospora. Figure 1. Life cycle of yeast. Problems based on mapping. Here, we're using an B loci for convenience. When an ab spore gamete fuses with an A plus B plus spore and the diploid then undergoes meiosis, the spores can be isolated and grown as haploid colonies which are then observed for the phenotypes the two loci control. Only 3 patterns can occur. Figure 2. Parental to type white circle. 2 different types of genotypes in tetrad and both of them must be parental types. White circle only parental or non-recombinant. Ascospores white circle no crossing over. Non-parental to type white circle only non-parental or recombinant. Ascospores white circle represents the unlinked segregating loci. Tetratype white circle contains 4 different types of genotypes out of which 2 are parental types and the other 2 are recombinant ones. White circle indicates single crossing over between 2 linked loci. Figure 2. The pattern of segregation in a tetrad. Mapping problem. All 3 can be generated whether or not the 2 loci are linked. Table 1 1. Parental to type 2, non-parental to type 3. Tetratype ab plus ab. Ab plus ab plus. A plus B plus A plus B A plus B. A plus B plus A plus B A plus B plus 7 5 5 2 0 table 1 data of tetrad analysis. Calculating the map distance between the loci. In the given problem, the PD greatly outnumbers the NPD. The 2 loci are, therefore, linked. In NPD, all 4 chromatids are recombinants and in TT, only half the chromatids are recombinants. 1 the number of TT number. Of NPD 2 map units 100. Total number of a C. Plus. Equals times. 1205. 2 map units 100. 100. Plus. Equals times. Ordered spores in Neurospora life cycle. Figure 3. Figure 3. Life cycle of Neurospora. Meiosis in Neurospora is followed by one more round of mitosis which yields 8 ordered ascospores in ascus. Figure 4. Figure 4. Optic formation in Neurospora. Ordered tetrad. The arrangement of spores in Neurospora crassa is very tight and they remain in a narrow ascus. Hence, after meiosis they produce ordered tetrads. Because of the precise positioning of each meiotic product within the ascus, one can infer the arrangement and segregation of each chromatid of homologous chromosomes during meiosis I and II. The distance between the centromere and the gene can be estimated, as the tetrad is linearly arranged. Meiosis II is followed by mitosis resulting in each pair of genetically identical daughters sitting adjacent to one another. Tetrad, then, is converted into octet 8 haploid ascospores. First division segregation FDS and second division segregation SDS. The segregation of 2 alleles of respective genetic loci can be done in the first or second division of meiosis. Conditions. White circle if no crossing over occurs between the genetic locus of allele and centromere. They demonstrate. Black small square first division segregation FDS Fig. 5 white small square migrating away from each other in meiosis I. Figure 5. First division segregation. White circle if a single crossing over occurs between the genetic locus of allele and centromere. They demonstrate. Black small square second division segregation SDS Figure 6. White small square migrating away from each other in meiosis II. Figure 6. Second division segregation. These conditions allow one to locate the gene on the chromosome if there is a correlation between SDS and the genetic distance between gene and centromere. Spore arrangement shows that crossing over takes place at 4-strand stage. Figure 7. Figure 7. Spore arrangement shows that crossing over takes place at 4-strand stage. No crossover between the centromere and the gene or a crossover involving two of the 4 strands results in a change in the arrangement of the spores. Figure 8. Figure 8. The arrangement of the spores when no crossing over occurs. An arrangement of the spores when crossing over between A plus and centromere occurs B is shown in this figure. Consequences of two crossovers between the same two strands and B between two different strands, shown in figure 9. Figure 9. Consequences of two crossovers between the same two strands. 2-strand double crossover B between two different strands. 3-strand and 4-strand double crossover. Detection of linkage. Spore order. Ascus number. 1 2 3 4 5 6 7 8 9 1 0 1 8 8 8 8 2 8 8 8 3 8 8 4 8 8 5 8 6 8 8. Mapping with molecular markers. Molecular markers. A molecular marker is a segment of DNA found at a specific site along a chromosome and has properties that enable it to be uniquely recognized using molecular tools such as PCR and gel electrophoresis. Molecular markers may be polymorphic, that is, they may vary from individual to individual. Distances between linked molecular markers can be determined from the outcomes of crosses. For both ordered and unordered spores, linkage is indicated by an excess of PD over NPD. For unordered spores, the distance between two loci is 1/2 the number of TT plus the number of NPD, all divided by the total number of C, expressed as a percentage. For ordered spores, neurospora, the distance from the locus to its centromere is 1/2 the percentage of SDS. Mapping the distance between two loci is similar for unordered spores. Chapter summary. Common types of molecular markers. Marker description RFLP restriction. Fragment length. Polymorphism. Differences in homologous DNA sequence can be detected by the presence of fragments of different lengths after digestion of the samples in question with specific restriction enzymes endonucleases RE. AFLP amplified fragment length polymorphism DNA fragments 80 to 500 BP are obtained from digestion with 3, followed by ligation of oligonucleotide adapters to the digestion products followed by pre-amplification and selective amplification by PCR. STR short. Tandem repeat. It is a pattern of 2 to 6 nucleotides that are repeated directly adjacent to each other. The total length usually ranges from 100 to 150 BP. An STR polymorphism occurs when homologous STR loci differ in the number of repeats between individuals. SNP single nucleotide. Polymorphism. It is a site where a single nucleotide is polymorphic among different individuals. These sites occur commonly in all genomes and they are gaining greater use in the mapping of genes that contribute to quantitative traits that are valuable in agriculture. STS sequence tag site. It is a general term to describe any molecular marker that is found at a unique site in the genome and is amplified by PCR. AFLP, microsatellites, and SNPs can provide STS sites within a genome. Table 4. An RFLP probe is a labeled DNA sequence that hybridizes with one or more fragments of the digested DNA sample after they have been separated by gel electrophoresis, thus revealing a unique line pattern characteristic of a specific genotype at a specific locus. Short, single or low copy genomic DNA or cDNA clones are typically used as RFLP probes. The RFLP probes are frequently used in genome mapping and variation analysis, genotyping, forensics, paternity tests, and hereditary disease diagnostics. RFLP method. The difference in homologous DNA sequences can be detected by the presence of fragments of different lengths after digestion of the DNA samples in question with specific restriction endonucleases. Figure 10. RFLP, as a molecular marker, is specific to a single clone restriction enzyme combination. Most RFLP markers are codominant. Both alleles in a heterozygous sample will be detected and highly locus specific. Figure 10. RFLP. RFLP. Genotyping. Figure 11. Figure 11. Genotyping by RFLP. Total DNA is digested with a methylation site of enzyme, for example, PSTI, thereby enriching the library for single or low copy expressed sequences. PSTI clones are based on the suggestion that expressed genes are not methylated. The digested DNA is size fractionated on a preparative agarose gel in fragments ranging from 500 to 2000 BPR size, eluted, and cloned into a plasmid vector, for example, PUC18. Digested the plasmids are screened to check for inserts. Southern blots of the inserts can be probed with total shear DNA to select clones that hybridize to single and low copy sequences. The probes are screened for RFLPs using genomic DNA of different genotypes digested with restriction endonucleases. Typically, in species with moderate to high polymorphism rates and 2 to 4 restriction endonucleases are used such as E. cori. AFLP amplified fragment length polymorphism. AFLP PCR was first described by researcher Peter Voss and his colleagues in 1995. Voss et al. 1995. This technique involves 5 major steps. White circle preparing the AFLP template. White circle ligation reaction with restriction. Fragments and adapters white circle selective PCR amplification. White circle electrophoretic separation of amplified. DNA fragments white circle analysis. AFLPs are differences in restriction fragment lengths caused by SNPs or indels that create or abolish restriction endonuclease recognition sites. The AFLP technique is based on selective PCR amplification of restriction fragments from a total digested genomic DNA. Figure 12. Figure 12. AFLP. AFLP permits high resolution interrogation of the entire genome. It yields highly specific, reproducible genotypic data. Although AFLP can only be used to study dominant genetic markers, it does not rely on any previous knowledge of the genome sequence. AFLP. How to use AFLP data. This technique can be especially useful in addressing questions related to organisms whose genome sequence has not yet been determined, including many plants, fungi, and bacteria. AFLP can help indicate whether two organisms are members of the same species. It can be used to assess genetic variation within a species or among closely related species. Population geneticists also use AFLP approaches to determine genetic variation across different populations. This method has allowed researchers to refine the taxonomic classification of organisms based on AFLP-associated genetic markers. AFLP can also be used to study organisms whose genome has been sequenced. For example, AFLP has been used to study human DNA samples in criminal investigations and paternity tests. In the hospital setting, AFLP has been used to track pathogenic outbreaks to determine whether an outbreak is associated with the spread of a single strain or multiple distinct strains. In this type of analysis, the pathogenic organism is isolated from infected patients and subjected to AFLP analysis. By comparing AFLP data collected from different patients, researchers can readily determine whether the outbreak stems from one or many different strains. Summary of molecular markers. Figure 13. Figure 13. Different types of molecular markers and their properties. Mapping by using somatic cell hybrids. The process of in vitro fusion of protoplasts derived from a somatic cell of either the same or two genetically different organisms is called somatic hybridization. Figure 14. Figure 14. Fusion of two somatic cells. It is used to assign a gene to a particular chromosome. For e.g., if human and mouse cells are mixed in the presence of PEG polyethylene glycol, fusion results in human mouse somatic cell hybrids. Figure 15. The hybrid cells tend to lose chromosomes as they divide. Generally, chromosomes of one species are lost preferentially, unknown reason. In human mouse somatic cell hybrids, human chromosomes tend to be lost. The chromosome number stabilizes when all except a few of the human chromosomes have been lost. Chromosome loss is random and differs among cell lines. The presence of extra human chromosomes in the mouse genome makes it possible to assign human genes to specific chromosomes. Figure 15. Somatic cell hybridization. Mapping of genes via somatic hybridization. Requires a panel of hybrid cell lines. The cell lines of the panel are chosen so that they differ in the human chromosomes that they have retained. For EG, one cell line might possess 1, 2, 4, and 5, whereas another might possess 2, 17, and 22. The human gene can be detected by white circle of the gene itself tagging PCR or white circle of the gene product. Correlation of the presence of the gene with the presence of specific human chromosomes often allows the gene to be assigned to the correct chromosome. For EG, if a gene is detected in both of the aforementioned cell lines, the gene must be present on chromosome 4 because it is the only human chromosome common to both cell lines. The basis of somatic cell hybridization. Protoplast fusion. Formation of viable heterokaryon. Fusion of nuclei and the division of hybrid cells and their subsequent growth. Identification, isolation or selection of hybrid cell. Selection of hybrids. After fusion, both homokaryon and heterokaryon will be present. During selection, one must prevent the growth of parent cells and permit the growth of hybrid cells. Materials: white circle non-toxic fluorochromes, fluorescein isothiocyanate fit, or rhodamine isothiocyanate (RITC). White circle for recovery, hat medium. White circle biochemical and molecular techniques such as isoenzyme pattern, RFLP and PCR can also be used. Gene mapping is a very important study of genetics that can resolve many problems that are encountered in daily life. The mapping of the genome can be of various types and some have been discussed in this chapter. Tetrad analysis is used for haploid organisms. Chapter summary. 5 extra chromosomal inheritance. Other proteins for specific roles and their metabolism. Cytoplasmic inheritance. The transmission of genes occurring outside the nucleus is termed as extranuclear or unaparental or cytoplasmic inheritance. It is found in most eukaryotes and is commonly known to occur in cytoplasmic organelles such as mitochondria and chloroplasts or from cellular parasites like viruses or bacteria. Mitochondrial molecular biology. In cell division, the mitochondrial genome segregates into progeny cells at random. The probability of any cell receiving mostly mutated mtDNA or wild-type mtDNA is random and chance-based. A mitochondrion is divided into two in the G1 phase of the cell cycle, simply by splitting or fission. Figure 1. Introduction. Mendelian and extensions of Mendelian factors are related to the inheritance of nuclear genes. However, some non-Mendelian patterns of inheritance are also seen in living beings. Extrachromosomal inheritance is referred to as the inheritance governed by the genes present outside the nucleus. Such type of inheritance is mostly found in eukaryotes. It is commonly observed in cellular parasites, viruses, bacteria or protozoans, and organelles like mitochondria and plastids. These cytoplasmic inheritance patterns were first reported by Carl Correns 1908 in plastids of Morales jalapa that do not follow Mendelian inheritance. Cytoplasmic inheritance is also termed maternal or unaparental inheritance because the cytoplasm is only contributed by the female gamete. Extrachromosomal genome contains a few genes mainly encoding rRNA. Turn sum. Figure 1. Division of mitochondria by splitting or fission. General patterns of inheritance in mitochondria. Mitochondria are generally inherited in a maternal pattern. Male gametes don't participate, but a small amount of leakiness is found in the process. White circle for EG. Mice have 1/1000th of the paternal mitochondria. White circle in mussels. Mitochondrial inheritance is biparental. White circle in gymnosperms, coastal redwoods, inherited paternally. Mitochondrial inheritance is homoplasmy, the existence of a uniform population of mitochondria within an organism. White circle biparental inheritance and leakiness show heteroplasmy, heterogeneity of mitochondria. Figure 2. Figure 2. Heteroplasmy in mitochondria. Mitochondrial genome, chondriome. The size of mtDNA in different organisms is termed mitochondrial genome or chondriome provided in table 1. Genome size, small and varies in different organisms. SN organism size KB. 1. Homo sapiens sapiens 16.6. 2. Mus musculus 16.2. 3. Xenopus laevis 18.4. 4. Drosophila melanogaster 18.4. 5. Saccharomyces cerevisiae 75. 6. Pisum sativum 110. 7. Arabidopsis thaliana 367 table 1 mitochondrial genomes of various organisms. Derived, found in most animal and fungal mitochondria. White circle smaller than ancestral and contain fewer genes. White circle DNA sequence differs from the eubacterial ancestors. White circle they contain non-universal codons. Table 2. Human mtDNA. Figure 3. Circular 16,569 BP. Encodes 2 rRNA, 22 ternus, and 13 proteins. 2 nucleotide strands differ in base composition. White circle heavy H strand. More G content. Right pointing arrow template for both rRNAs. 14/22 terna and 12/13 proteins. White circle or right pointing arrow for each strand is within D loop. Triple stranded structure. White circle light L strand. More C nucleotides. Right pointing arrow 8. Ternus and 1 protein. No introns. Each strand right pointing arrow single promoter. Right pointing arrow very large single RNA is synthesized, which is later cleaved into individual RNAs. Many genes lack complete termination codon. Either U or UA tail is present. D loop contains some non-coding nucleotides. Repetitive sequences and some duplication are not found. Multiple cDNA. Plant mitochondria genome. Histone proteins normally associate with mtDNA. GC content is different from nuclear DNA. Hence, it can be separated from nuclear DNA by density gradient centrifugation. Size variation in intergenic intron regions. Gene structure and organization of mtDNA. The genes for many of the structural proteins and enzymes found in mitochondria are encoded by nuclear DNA, translated on cytoplasmic ribosomes, and then transported into the mitochondria. The mitochondrial genome typically encodes only a few rRNAs and internal molecules needed for mitochondrial protein synthesis. Ancestral and derived mitochondrial genome. The genome can be subdivided into ancestral and derived. Ancestral, found in some plants and protists and retain many characteristics of eubacterial ancestors. White circle contain more genes as compared to derived ones. White circle have rRNA genes and a completer. Almost complete set of Turner genes. White circle possess few introns and little ncDNA. White circle genes are organized in clusters. Figure 3. Mitochondrial genome. Mostly, the extra region is non-coding. Genes encoding polypeptides include regions that encode 5' and 3' UTR of mRNA. Repetitive sequences and some duplication are also found. Yeast mtDNA. Figure 4. Circular 78,000 BP, 5 times larger than human's mtDNA. Encodes 2 rRNA, 25 ternus, and 16 proteins. Only additional 6 genes. Figure 4. Human and yeast mtDNA. Flowering plant mtDNA. Angiosperms have the largest and the most complex mitochondrial genomes known. 1. 86,000. White mustard to 24. OOBP. Muskmelon. Figure 5. Reason: the presence of large direct repeats. Crossing over between these repeats generations of multiple circular chromosomes of different sizes. Figure 5. Mitochondrial and plastid genome of a flowering plant Arabidopsis thaliana. Codon universal code. mtDNA. Vertebrates Drosophila yeast. UGA stop tryptophan tryptophan AUA isoleucine methionine 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 other arginine serine serine arginine. Table 2 non-universal codons in mtDNA. Replication, transcription, and translation in mtDNA. Replication. White circle does not replicate in an orderly, regulated manner like the nuclear DNA and is synthesized throughout the cycle. White circle the mtDNA molecules that are replicated at any particular moment are random, within the same mitochondrion. Some molecules are replicated twice or thrice, whereas others are not replicated at all. White circle unidirectional replication. White circle replication occurs asynchronously in 2 strands, replicated by DNA pole gamma. Transcription. White circle in humans, 2 promoters 1 each 4. H and L are present within the D loop. White circle transcription starts bidirectionally, right pointing arrow generating 2 giant RNA precursors, right pointing arrow turna, rRNA, and mRNA are then cleaved from those. White circle in fungi, plants, and protists, multiple promoters, right pointing arrow genes organized in clusters operons. White circle mRNA, right pointing arrow not capped and 3' poly tail is added but is shorter than nuclear mRNA tail is missing in fungi, plants, and protists. White circle some plants and yeast mtDNA contain self-splicing introns. White circle RNA shows extensive editing. Translation. White circle translation starts at AUG by informal methionine. White circle similar to bacterial protein synthesis. It is inhibited by chloramphenicol prokaryotic protein inhibitor. White circle animal mtDNA. No shine Delgarno SD. Sequence in no 5' cap. White circle in a protozoan briefly known as Americana. The SD sequence is observed. White circle more wobble turn at 22 as compared to cytosolic 3-2 turnus. White circle more changes in mtDNA are silent and accumulated right pointing arrow higher rate of evolution. Evolution of mtDNA. A common bacterial origin for all invertebrates. Accelerated rate of change in plants. The rate of evolution is slow. Mitochondrial inheritance in yeast. Yeast is a facultative anaerobic organism. Yeast cells growth in white circle aerobic conditions. Distinctive colony. Morphology white circle anaerobic conditions. Smaller colonies in mitochondria are shorter. Figure 6. The segregational petite. White circle caused by mutation of a chromosomal gene exhibits Mendelian inheritance. The neutral petite mutagen acridine orange ethylene bromide ETBR. White circle is lost immediately upon crossing to the wild type. The suppressive petite mutagen acridine orange ETBR. White circle shows variability in expression from one strain to the next but is able to convert the wild type mitochondria to the petite form. Petite, failure in mitochondrial function. They usually lack one or another cytochrome. Figure 6. Normal and petite colonies of yeast drawing. Petite in yeast. These colonies are caused by petite mutations. When petite is crossed with wild type, 3 modes of inheritance emerge. Figure 7. Figure 7. Petite in yeast. Toki neurospora. Figure 8. Toki mutants grow slowly and have abnormal amounts of cytochromes. They have cytochrome C but not cytochrome RB. Figure 8. Inheritance in Toki neurospora. Male sterility in maize. Figure 9. Male sterility in maize follows 2 patterns. White circle one in which trait is inherited through a single recessive chromosomal gene segregating in Mendelian ratios. White circle those which show maternal inheritance. Figure 9. Male sterility in maize. White circle the maternal inheritance of mtDNA. Chloroplast molecular biology. Chlorophyll containing organelle carries out photosynthesis. Referred to as plastids before chlorophyll develops. In dark, plastids do not develop into chloroplasts. Term proplastids reduce size and complexity. Chloroplasts generally show homoplasmy and maternal inheritance like mitochondria. Gymnosperms usually have paternal inheritance. Chloroplast DNA plastome. Genome size ranges from 80,000 to 6,000 Table 3. Single DSC DNA, highly coiled and lacks associated histone proteins. Multiple copies of the chloroplast genome are found in each chloroplast and multiple organelles per cell. Human mitochondrial inheritance. Some diseases. Luff disease. Labor optic atrophy or neuropathy. Myoclonic epilepsy and ragged red fiber disease syndrome, caused by mutation in the ternagene. Neurogenic muscle weakness, ataxia, and retinitis pigmentosum, NAP. Kearns-Sayre syndrome, KS, chronic external ophthalmoplegia, CEOP. Mitochondrial DNA variation in human history. mtDNA is studied extensively to reconstruct the evolution and migration patterns of humans. Advantages of mtDNA for studying evolution are: white circle, the small size and abundance of mtDNA. In the cell white circle, the rapid evolution of mtDNA sequences facilitating the study of closely related groups. SN organism size of cpDNA BP. 1. Euglena gracilis 143,172. 2. Porphyroporphyria 150,613. 3. Chlorella 191,028. 4. Marchantia polymorpha 121,024. 5. Nicotiana tabacum 155,939. 6. C. maize 140,387. 7. Pinus thunbergi 119,707. Table 3 sizes of chloroplast genomes in selected organisms. Gene structure and organization of cpDNA, figure 10. In eubacteria, genes are organized in operon-like clusters. In plants, similar in gene content and gene order. Encodes 4 rRNA genes, from 30 to 35 turnigenes, a number of ribosomal proteins white circle involved in photosynthesis and other processes. Circular CP genome has genes on both of its strands. Prominent feature of cpDNA genes, the presence of a large inverted repeat ncDNA and introns are present. Figure 10. Chloroplast genome. Replication. Replication begins within 2D loops and spreads outwards to form a theta-like structure. After an initial round of replication, DNA synthesis may switch to a Rowland circle amplification mechanism. Transcription and translation. Similar to eubacteria, however, introns are removed after transcription and may require some post-transcriptional processing too. Larger ribosome units include 3 rRNA molecules, a 23 srRNA, a 5 srRNA, and a 4.5 srRNA, 23 srRNA, a 5 srRNA in eubacteria. 4.5 srRNA is homologous to the 3' end of the 23 srRNA found in eubacteria. Only universal codons are found in cpDNA. Evolution of cpDNA, figure 11. The DNA sequences of chloroplasts are very similar to those found in cyanobacteria. Eubacterial ancestry, figure 11. cpDNA evolves slowly as compared to nuclear and some mtDNA. Figure 11. Evolution of mitochondrial and chloroplast. Movement of genetic information between nuclear, mitochondrial, and chloroplast genomes. Some sequences normally found in mtDNA have been detected in nuclear DNA. Gene flow is seen from chloroplast to mitochondria as well as nuclear to mitochondria. Horizontal gene transfer, referring to transfer from one plant to another, is also seen in mtDNA. Leaf colored in Mirabilis jalapa. In 1909, Corrin studied the inheritance of leaf variegation in the 4 o'clock plant.